Good morning, students. Welcome you all in our e-learning session of classroom. In our today's lecture, we are going to learn the remaining part of from the chapter number 5. Yesterday we start the chapter number 5, that is origin and the evolution of life. In that chapter or in that lecture, we learn the two experiment. First one, the Urey and the Miller experiment. In that experiment, the scientists took 1:2:2 proportion, that is methane, ammonia and hydrogen gas in 1:2:2 proportion. They used the glass apparatus known as a spark discharge apparatus and continuously light electrical supply is provided to that apparatus, and from that experiment they concluded that here the gases contains and brown colored ppt or liquid is formed in that U2. Next we learn the second part, that is RNA World Hypothesis. In that RNA World Hypothesis, again, we know that RNA, that is the ribonucleic acid, which is very important in the process of, which is present in all living organisms. And the ribozyme, which is, ribosomes are considered as the protein unit. That said, for the synthesis of protein, ribosomes are important. And here we know that the central dogma from DNA, transcription, mRNA, from mRNA, protein with the help of the translation. Here the RNA World Hypothesis is play very important role in all living organisms. In our today's lecture, we are going to learn about the next part from the same chapter, that is the modern synthetic theory of evolution. Okay, in that modern synthetic theory of evolution, it is a result of true synthesis of all biological disciplines. Okay, studied pertaining to genetical, ecological, anatomical, geographical and the paleontological, etc. were pursued to explain the mechanism of evolution. Also, due importance was given to both mutation and the natural selection. Okay, here some experiment is taken place by these scientists. They are main contributor of the modern theory of evolution. And in such a way that 5 key factors such as gene mutation, mutation in chromosome structure and the numbers, genetic recombination, natural selection and the reproduction isolation contributed in the evolution of new species. Okay, now all individuals of the some species constitute a population. As we know that all individuals of the some species, every species has its own population and they contribute its own population in that particular species. All individuals of the some species constitute a population. The population occur in small group of interbreeding population. Here the interbreeding population. Such small interbreeding group of population is referred as Mendelian population. Here the population occur in small group, that is the interbreeding. त्यांच्यामध्ये होणारं ब्रीडिंग जे आहे त्याला काय म्हणतात, interbreeding population. Such small interbreeding group of population is referred as Mendelian population. Okay, the total genetic information encoded in sum total of the total genetic information is encoded in sum total of gene in Mendelian population It's called as gene pool. What is gene pool? Here the gene pool is maybe asked for the definition. Define the gene pool. The total genetic information encoded in sum total of genes in an Mendelian population is called as gene pool. Okay. Simply, gene pool means the total number of genes of all individual in the population. म्हणजे त्या population मध्ये असणारे जे काही individuals आहेत त्यांच्यामध्ये total number of genes त्याला काय म्हटलं जातं gene pool. Okay, simply त्यांनी सांगितले gene pool means total number of genes of all individual in a population. The gamete produced by individual furnish a pool of gene of next generation from which the gene will selected. Okay, I repeat. The gametes, male gamete, female gamete, produced by individuals, male and the female, furnish a pool of gene of next generation, produced for the next generation, from which the gene will be selected. And त्याच्यातलेच superior quality gene काय केले जातात? Select केले जातात. The migration of population effectively alter the gene pool. And ह्या ठिकाणी important migration, transfer from one place to another place for the food, shelter, etc. The migration of population effectively alter the gene pool. Gene pool also change due to replacement of one generation by another in the Mendelian population. The migration of population effectively alter the gene pool. Okay, the gene pool also changes due to replacement of gene pool. आणखी काय change केला जातात? Due to the replacement of one generation by another. एका generation साठी कडून दुसऱ्या generation द्वारे in the Mendelian population. कुठे? त्या Mendelian population मध्ये. Thus, any change in the gene pool affect population. आणि ह्याचाच रिझल्ट आपल्याला काय पाहायला मिळतो? Any change in gene pool affect the population. आणि ह्या ठिकाणी जर अशा प्रकारे काही मायग्रेशन द्वारे जर रिप्लेसमेंट झाली आहे, नेक्स्ट जनरेशन मध्ये त्या ठिकाणी तर काही प्रॉब्लेम झाला असेल किंवा रिप्लेसमेंट द्वारे चेंजेस ऑफर असेल तर ते सर्व चेंजेस जे आहेत ते त्या जीन पोलच्या पॉप्युलेशनला अफेक्ट करतात. ओके, नाउ genes are arranged linearly on the chromosome having definite position. Okay, where genes are present, as we know that nucleus cell, in that cell cytoplasm and nucleus are there. In cytoplasm number of cell organelles are there. In nucleus there are nucleoplasm, nucleolus, and here the genes, chromosomes are present in that particular nucleus. Genes are arranged linearly on the chromosome. Means genes are arranged on the chromosomes. Chromosomes carry the genes having definite position and they have their definite position. According to Mendel, every gene that influences a trait has two alleles. I repeat, according to Mendel theory, we know that the Mendel theory or Mendel is a scientist. Here, every gene, what Mendel says, every gene that influences a trait has two alleles, two characters. The proportion of an alleles in the gene pool to the total number of allele at given locus is known as or is called as gene frequency. Again, gene frequency is important. It will be asked for 1 mark question or define the gene frequency. What is gene frequency? Every gene that influences a trait has two alleles. The proportion, here from here the definition starts. The proportion of an allele in the gene pool to the total number of allele at a given locus is called gene frequency. Okay. Modern synthetic theory comprises 5 main factors that are broadly divided into three main concepts. Like first one, genetic variation caused due to the various aspects of mutation, recombination, and the migration. Here, the modern synthetic theory gives the five factors. These five factors are divided into three main concepts. Which three main concepts? First one is the genetic variation. Genetic variation, vividata. Genetically differences caused due to various aspects. कशामुळे होते? Due to the mutation, that is a sudden change. Recombination. दोन कॅरेक्टर्स मध्ये पुन्हा एकदा कॉम्बिनेशन केल्यानंतर डबल कॉम्बिनेशन रिपीट झाल्यानंतर कॉम्बिनेशन अँड मायग्रेशन ट्रान्सफर फ्रॉम वन प्लेस टू अनदर प्लेस. ओके, मायग्रेशन. सेकंड, नॅचरल सिलेक्शन. द सेकंड aspect is the second concept is the natural selection, and the third one is isolation for explaining the evolution of species. Isolation for explaining the evolution of species. Now, the first one is genetic variation. Okay, the change in gene and gene frequency is known as genetic variation. What is genetic variation? The change in gene and gene frequency. Genes मध्ये and gene frequency मध्ये जर काही change असेल तर त्यांना काय म्हणतात? Genetic variation. Genetic variations are caused by following factors. Here are some factors which cause the genetic variation. What these factors are? What the factors are? First, gene mutation. Sudden change. Sudden, permanent, heritable change is called as mutation. What is mutation? Sudden, अचानक झालेला, permanent, हेरिटेबल की जो एका जनरेशन कडून दुसऱ्या जनरेशन आपण कॅरी फॉरवर्ड करू शकतो. चेंज इन इज कॉल्ड म्युटेशन. म्युटेशन कॅन ऑकर इन द जीन, in the chromosome and in chromosome number. Mutation that occur within a single gene is called point mutation or gene mutation. What is point mutation or gene mutation? Mutation that occur within a single gene. एका particular gene मध्ये झालेले जे काही changes आहेत की म्युटेशन आहे त्यांना काढलं त्या पॉइंटला झालेलं म्युटेशन किंवा पॉइंट म्युटेशन और जीन म्युटेशन. ओके, this lead to the change in the phenotype. This lead to the change in the phenotype of the organism, causing what is called variation. Okay, this lead to the change in the phenotype. What is phenotype? Phenotype is external character. For example, if we consider organism, the external character. If we consider a plant, the green color leaves, red color flowers. If we consider an animal, that is the color of that animal, structure of that animal. These are called as the phenotype, the external characters. Okay, this phenotype, this lead to the change in the phenotype of organism, causing what is called variation. आणि त्याच्यामुळे आपल्याला व्हेरिएशन जे आहे ते स्पीसीज टू स्पीसीज पाहायला मिळतं. सडन चेंज झाल्यामुळे एकाच जनरेशन मधून दुसऱ्या जनरेशनकडे जाताना जीन मध्ये काही चेंजेस झाल्यानंतर, म्युटेशन झाल्यानंतर ऑटोमॅटिकली आपल्याला व्हेरिएशन पाहायला मिळतं. आणि याचं बेस्ट एक्झाम्पल जर आपण पाहिले तर आपण सख्खे भाऊ बहीण देखील जर पाहिले तर त्यांच्यामध्ये आपल्याला वेगवेगळेपणा व्हेरिएशन पाहायला मिळतं. का वेगळेपणा ते व्हेरिएशन असतं? जरी त्यांचे आई वडील एक असेल तरी सख्या भाऊ बहीण वेगवेगळे दिसतात. का? कारण त्याच्यामध्ये जीनचे जी म्युटेशन आहे ते म्युटेशन पाहायला मिळतं, चेंजेस पाहायला मिळतात आणि रिकॉम्बिनेशन पाहायला मिळतं आणि त्यामुळे व्हेरिएशन्स जे आहेत ते व्हेरिएशन्स आपल्याला ऑर्गॅनिझममध्ये पाहायला मिळतात. जेनेटिक रिकॉम्बिनेशन, द सेकंड टाइप or second concept is genetic recombination. In sexually reproducing organism, during gamete formation, exchange of genetic material occur between non-sister chromatid of homologous chromosomes. This is called crossing over. Again, this is important in case of definition. What is crossing over? In sexually reproducing organism, during gamete formation, male gamete, female gamete. Male gamete, in case of human being, we know that the male gamete sperms, testes produces sperms, and the ovaries produces ova. Male gamete and female gamete. Gamete formation takes place. Exchange of genetic material. Genetic material काय केलं जातं त्या ठिकाणी exchange केलं जातं. Occur between non-sister chromatid. We know that there are 23 pairs of chromosomes are there in human being. The last pair, that is the 23rd pair of chromosome, is responsible for the sex determination, or it is called as sex chromosome. This sex chromosome or the organism, this sex chromosome or the 23rd pair of chromosome is responsible for the male or female which kind of organism will be or offspring will be produced. It is the male or the female is decided by the 23rd pair of chromosome. Here the non-sister chromatid of homologous chromosome, this is called as crossing over. The chromosomes or the chromatids are crossing over on each other, and here the genetic material exchange. Okay. It is produce new genetic combination. आणि या ठिकाणी काय केलं जातं नवीन जेनेटिक कॉम्बिनेशन तयार केलं जातं. मेल गॅमेट आणि फिमेल गॅमेट एकमेकांमध्ये इंटरचेंजेस होतात, त्यांच्यामध्ये एक्सचेंज केलं जातं आणि ह्या एक्सचेंजसाठी या ठिकाणी क्रॉसिंग ओव्हर इम्पॉर्टंट आहे. It produces new genetic combination which result in variation. Why variations is occur or where it is occur? During the crossing over, and here it produces the genetic combination, and here we known as the genetic recombination. नवीन combination चे genes जे आहेत त्या ठिकाणी आपण पाहतो. Fertilization between the opposite mating gamete leading to various recombination resulting into the phenotypic variation causing changes in the frequency of the alleles. And this is another concept or the factor which is affect the variations or changes in the frequencies. Third one is the gene flow. Gene flow is movement of gene into or out of population. Okay, what is gene flow? Gene flow is the movement of genes. एक है gene movement की जे एक तर आतमध्ये जाऊ शकते किंवा बाहेर जाऊ शकते त्या population. Gene movement may be in the form of migration of organism or gamete dispersal of pollen or the segments of DNA, that is the transformation. Gene flow also alter the gene frequency, causing the evolutionary changes. Also, this causes the evolutionary changes. Now, the next one is the genetic drift. Any random fluctuation, that is the alteration in allele frequency occurring in the natural population by pure chance is called as genetic drift. I repeat, any random fluctuation or alteration, random, in allele frequency occurring in natural population by pure chance is called as genetic drift. For example, when the size of a population is severely reduced due to natural disaster like earthquake, floods, fires, etc., causing elimination of particular allele from a population. Smaller population have greater chances for genetic drift, and it will result in the change in the gene frequency. Genetic drift is also an important factor for evolutionary change. Genetic drift is important factor for the evolutionary change. Why? Because when the size of population is severely reduced due to the natural disaster, एखादी population ची size तिथले असणारे numbers जे आहेत ते जर naturally disaster मुळे, for example earthquake असेल, floods असेल, fire असेल, त्याच्यामुळे जर कमी झालं तर त्याच्यामुळे काय होतं, causing elimination of particular allele. आपल्याला माहिती आहे कमी झालं म्हणजे काहीतरी एलिमिनेट होते, त्यातलं पर्टिक्युलर अलिल्स जे आहे ते पॉप्युलेशन मधून कमी होतात. स्मॉलर पॉप्युलेशन हॅव ग्रेटर चान्स फॉर द जेनेटिक, आणि त्या ठिकाणी राहिलेले स्मॉलर पॉप्युलेशन आहे त्यांच्यामध्ये ग्रेटर चान्स असतात की ते त्या ठिकाणी काय करू शकतात, जेनेटिक ड्रिफ्ट करू शकतात. ओके, आणि इट विल बी रिझल्ट इन चेंज इन द जीन फ्रिक्वेन्सी, आणि त्याचाच रिझल्ट म्हणजे काय की त्या ठिकाणी जीन फ्रिक्वेन्सी आपल्याला चेंज झालेली पाहायला मिळते. आणि जेनेटिक ड्रिफ्ट इज अन इम्पॉर्टंट फॅक्टर फॉर इव्होल्युशनरी चेंजेस, आणि या ठिकाणी जेनेटिक ड्रिफ्ट जे आहे एक इम्पॉर्टंट फॅक्टर आहे की ज्याच्यामध्ये चेंजेस जे ते पाहायला मिळतात. नेक्स्ट, क्रोमोझोमल ॲब्रिएशन. The structural morphological change in the chromosome due to rearrangement is called as chromosomal aberration. It changes the gene arrangement, order or sequence that result in the variation. Chromosomal aberration occur due to first deletion, means loss of gene from chromosome. Second, duplication. Genes are repeated or double in number on chromosome. Third, inversion. A particular segment of chromosome is broken and get reattached to the same chromosome in an inverted position due to 1800 twist. There is no loss for the gain or the gene complement for the chromosome. And the last one is translocation, transfer of location, transfer or transposition of a part of chromosome or a part or a set of gene to a non-homologous chromosome is called translocation. It is effected naturally by the transposons present in the cell. Okay, these are the four factors or the chromosomal aberration occur where the deletion, first one, duplication, deletion delete करणे, duplication double copy तयार करणे, inversion के याच्यामध्ये add करायचं, आणि last translocation के याच्यामध्ये आपल्याला transfer location change झालेले आपल्याला पाहायला मिळते. Okay, this is all about our today's lecture. In our today's lecture we learn the modern synthetic theory of evolution. In this modern synthetic of theory of evolution, here we learn the modern synthetic theory comprises 5 main factors that are broadly divided into 3 main concepts: genetic variation, natural selection, and isolation. In our today's lecture, we learn the genetic variation. In that genetic variation, we also learn the factors, that is the gene mutation, sudden change, okay. Second, genetic recombination, okay. Now, next we learn the gene flow, and next we learn the genetic drift, and last we learn the chromosomal aberrations. In that chromosomal aberrations, it is occurred due to the first deletion, duplication, inversion, and the translocation. This is all about our today's lecture. I hope you understood the modern synthetic theory of evolution. Okay, here the evolution is important. Some terminology or some factors, definitions, terms are important in case of definition. For example, gene pool, gene frequency. Okay, then the modern, these are the 3 concepts: genetic variation, natural selection, isolation. Again, what is gene mutation? This is important. What kind of changes occur in gene mutation? Genetic recombination. Okay, the question may be asked: write the different factors which are present in genetic variations, or you have to write these 5 factors, that is the gene mutation, genetic recombination, gene flow, genetic drift, and the chromosomal aberrations. Okay, these are the 5 factors which comes under the genetic variation. I hope you all understood our today's lecture. in our next lecture, we will learn the next part of our chapter, origin and the evolution of life. That is the next or the second part, that is the natural selection. In our today's lecture, we learn the genetic variation, natural selection and isolation. These three concepts are come under the modern synthetic theory. Okay, modern synthetic theory. Here, the gene pool, interpreting population, gene frequency, these are come under the modern synthetic theory of evolution. Okay, this is all about our today's lecture. Student, please prepare your own notes with the help of our textbook. And also to practice, prepare your own notes and study well. Okay, in our upcoming lecture we are going to learn about the next topic, and here stay home, stay safe. Okay, thank you.